So, after the discussion on this cell division, let me give you some numbers for you to realize that how fast or how slow a typical eukaryotic cells they grow. This slide actually shows you some of the numbers for some specific cells. Adult chondrocytes, the cell division doubling time is 24 to 48 hours. So, chondrocytes means this is the cartilage cells. Dermal fibroblast, there is cell division cell doubling time is 15 hours, whereas hematopoietic progenitors they typically double in 11 to 12 hours. Cardiomyocytes, which is the cardiac cells, these cardiomyocytes cannot be cultured in any meaningful period of time because it is very difficult to culture. So, clearly the cell division or cell doubling time depends on the cell type. Human osteoblasts, their division time cell doubling time is typically 48 hours, whereas fibroblasts they grow little fast 15 hours. Now, when you grow the cells in culture, one of the immediate parameters that researchers they use just to see that whether these cells grow in a predictable manner or not, that is they count the number of cells after growing them in different time scale like 4 days, 6 days, 8 days and so on, like over a week or more than a week. Now, that depends on what is the typical cell doubling time. Now, this slide shows you some of this data taken from different cell types, and mostly you can see that this cell growth actually is taking mostly the linear shape. So, essentially the number of cells that linearly grow with time in culture. This is some of the fluorescence microscopic images just to show the progress of cell division of a typical animal cell, and what you see that slowly the cells increase in size. You see that nucleus that all that significantly increases in size during the early stage of the cell division, and then there is a typical increase in this so increase in the cell nucleus is an indication that cell is preparing itself for the cell division, and slowly that during the different stages of the cell division, at the late stage you can see that the cytoplasm of one daughter cell is almost physically separated from another daughter cell, and this cytokinesis is the last step where the formation of the two daughter cells from a mother cell is complete. So, next thing I would like to start with, and I would like to complete in this particular module is the cell differentiation, and to this extent I will start with the typical description of the structure of DNA and RNA. Because as I said, differentiation means this is the differential gene expression. So, it is a kind of cell fate processes, it is one of the cell fate processes which involves differential gene expression. Now, to go back to my earlier description of a cell fate processes, when a cell adheres on a material substrate, the question that we need to address that whether the cell changes in cell fate processes, changes in cell fate. So, that means that whether the cell is functionality is changed, and the cell functionality changes can be assessed by certain differentiation assays and see that whether cell goes to a more mature cell type. Now, to understand the differentiation and to quantify the gene expression, you need to first start with the you need to first understand that what is the structure of two nucleic acids of importance, that is the D-ribonucleic acids and ribonucleic acids, DNA and RNA. So, these ones I have mentioned you before, that once a cell adheres on a material substrate, that what are the different fate processes that a cell can decide or cell can choose to take it up. Replication, that is the division, differentiation, death, cell motion and cell adhesion. So, I already mentioned that differentiation essentially means the differential gene expression. So, therefore, this involves one of the things that you must remember in the differentiation process, that involves the expression of unique genes that are more specific to the mature cell type. So, that means that you have one cell type, it is little bit premature, and one cell type which is much more mature cell type. So, genes which are specific to this particular mature cell type, that should be expressed, and that are specific to with an irreversible change towards a particular cellular function phenotype. So, the gene specific to the mature cell type, that should be expressed or that should be upregulated, and then only you can confirm, yes, a cell A undergoes differentiation to cell B, and certainly cell B will have different functionality compared to cell A. Okay. So, the third point that has been mentioned here, that is, it involves orchestrated switching off and on of certain gene families. So, these gene certain specific gene families which are switched on, that means that will be expressed now or that will be upregulated now, which will be related to the more mature cell type here. And final set of genes which are expressed or which are upregulated, that will pertain to the function of the differentiated mature cell itself. And like cell division process, differentiation is irreversible process. That means that once a cell goes from A to B, that is this initially this is irreversible in nature. Now, gene, what is gene? Gene is the nucleotide sequence in a DNA molecule that functions to synthesize protein, structure RNA and catalytic RNA molecules. So, essentially gene is defined as a nucleotide sequence in a deoxyribonucleic acid, and genome is that a complete set of information in an organism's DNA. Now, this information is undergoes transition or change from DNA to RNA, that means information is carried over from DNA to RNA. Organisms differ from one another because their respective DNA molecules have different nucleotide sequence and therefore carry different biological messages. And genes which carry biological information that must be copied accurately for transmission to the next generation each time a cell divides to form two daughter cells. That means once a cell goes to two daughter cells, so these genes are copied accurately. So, these genes from that mother cell is to be copied accurately to daughter cells each time the cell divides. A typical human cell contains 2-meter long DNA. 2-meter long DNA means you can understand that it is much longer than the height of even a tall person. That this long DNA, the DNA length, that means can be much longer than the height of a single person, but it is essentially squeezed in a nucleus in an extremely coiled form, and it also carries the instruction for 30,000 different proteins. Now, DNA structure, it appears often on the cover page of many standard biology textbook, and many of you have read the DNA structure in even in your school biology as well. So, this is a typical double helical structure, and DNA double helix structure was discovered quite some time back. Now, you can describe this structure in a more simplistic, or to start with you can describe this structure as two backbone chain. So, this is your two, so DNA is essentially made up of 4 type of nucleotides, A, C, G, T. So, adenine, cytosine, guanine and thymine, and this A is to always pair with T and C has to always pair with G, and this pairing is involved through some chemical bonding here. So, you have two DNA strand, this is strand number 1, this is strand number 2. Another thing you can see that these strand essentially replica of one another. So, these two long polypeptide chains are there, and along this chain you have the sugar phosphate and base, that is it forms a nucleotide here. So, the complementary base pairing, that is a very important thing in the DNA structure, enables packing in energetically most favorable arrangement. What it means that this double helical structure essentially represents the lowest energy configuration, because you know from basic thermodynamics any structure which in its equilibrium shape must represent the lowest energy configuration, then only that structure would be more stable. So, this is more elaborate view of this A, T, C, G. So, this is your A adenine, this is your thymine. So, you have these specific bonds here, and this is your C and this is your G. So, these are the two base pairing. So, this is one base pairing and this is the another base pairing, and these bonds are essentially very weak bonds. So, these bonds are hydrogen bonds. So, the advantages of having hydrogen bonds in this base pairing is that if required they can be simply removed, and therefore, these two strands can be separated easily, simply because these bonds which enable or which mediate the pairing between C and G or A and T are extremely weak, that is the hydrogen bonds. So, there are certain uniqueness of the DNA structure which has been summarized here. So, each strand in a DNA structure acts as a template, as I told you that it helps you synthesize new complementary strand. And so, if you designate S and S prime is two DNA strands, so strand S essentially serve as to template for the new strand, that is S prime, and the ability of each strand of a DNA molecule to act as a template enables a cell to copy or replicate its genes accurately down to two daughter cells. So, you have one mother cell, it goes to two daughter cells. So, essentially the DNA sequence here is to be exactly to be replicated in two daughter cells. Ribonucleic acid, their point of dissimilarity with respect to DNA structure is that in the ribonucleic acid in a DNA structure you have AT and then CG. In ribonucleic acid you have uracil in place of thymine. So, therefore, your A will base pair with U, that is uracil. So, one of the characteristics of a cell, eukaryotic cell, is that the DNA replication or DNA genetic recombination. So, from DNA you can DNA can undergo transcription process to form RNA. So, that takes place within the nucleus itself, and this is your cytoplasm. So, that is your nucleus. So, in the cytoplasm this RNA can undergo translation to form a protein molecule. As I have mentioned to you before, the protein in a given protein molecule, N terminal will always be kept at the left hand side and C terminal will always be kept on the right hand side. So, this DNA to RNA and RNA to protein. So, this first one is a transcription process and second one is the translation process. The way this was discovered at the early stage of this, at the early stage people used to think that this transcription and translation process is one way process. So, once DNA comes to RNA, RNA cannot go back to DNA, but later on people have found out that reverse transcription, that means that RNA also, the information contained in RNA can also go and change and form to DNA. So, this reverse transcription process is also possible, and therefore, this entire theory of transcription translation in the molecular biology, it is mentioned as the central dogma in the molecular biology theory. So, this is more mentioned here. This DNA transcription produces a single stranded RNA molecule, and that is complementary to one strand of DNA. What it means is that, so 5 prime 3 prime, and then it is complementary when it forms that RNA in the transcription process. And remember, in an eukaryotic cell, this transcription process always takes place well within the nucleus. So, this one has been shown here. So, you have different type of RNA. mRNA stands for messenger RNA, and it contains the code for proteins which will be ultimately synthesized in the cytoplasm of the cells, that is outside nucleus. Then you have ribosomal RNA, that is rRNA, and this actually basic structure of ribosome and catalyzes the protein synthesis. Then you have a transfer RNA, that is tRNA, that is central to protein synthesis, and also this essentially is also, this is one of that another RNA type which also takes part in certain biological processes. So, DNA to RNA, mRNA. So, mRNA is now comes to the cytoplasm through this nucleus pore complex, that NPC, which I have mentioned before. And then from mRNA it has two options, either it can be inactivated or it can be undergo translate, or it can undergo translation to synthesize protein in the cytoplasm. This entire thing has been shown here in the prokaryotic cells, that within the cytoplasm itself the transcription translation process takes place, simply because prokaryotes does not have very well defined nucleus within the bacterials within the prokaryotes. So, this one of the techniques that is used in the cell biology, it is a polymerase chain reaction. Now, what is this polymerase chain reaction? So, once a cell differentiates to another, or once a cell is committed to undergo differentiation to another cell type, then you have to extract mRNA from the cultured cells. Now, the quantity of mRNA can be extremely small. So, you have to amplify this mRNA expression, and this amplification takes place in using this amplification is possible if mRNA undergoes now polymerase chain reaction. So, it is essentially a technique which is used to amplify the number of copies of a specific region of DNA in order to produce enough DNA to be adequately tested. So, there is a detailed biology involved in discussing that PCR, but what is more important and more simplistic for you to realize, that you have the double stranded DNA. So, this is your template DNA. In the first cycle it forms 2 copies here. So, this essentially this is the 2 pairs of copies. So, the total copies is 4. In the second cycle, each of this copy will now produce another 4 copies. So, then second cycle essentially 4 4 8 copies that will form. In the third cycle, it again that each of this pair of the copies will produce again 4. So, it is to 16 copies finally, and likewise it will undergo exponential amplification, and then it would be 2 to the power n. So, if n is the total number of cycles, and then let us say if it undergoes 36 cycle, or at the end of 35th cycle, you have such a large number of copies like 68 billion copies that will be produced, which will be quite good enough to now quantify or to be quantified using this particular technique. So, this is that more PCR cycle, what they call the polymerase chain reaction. So, you first step is that step 1 is a denaturation. Denaturation takes place and these numbers are very specific, and this typically takes place at 94 degrees Celsius, below 100 degrees Celsius. Then something called annealing. The term annealing means it is one of the term which is used in material science, which is like heat treatment. So, essentially this heat treatment in the biology, it essentially takes place at a much slower time, much lower temperature like 54 degrees Celsius, and very short time period like 45 seconds, less than a minute. And then you have a extension, and then extension is 72 degrees Celsius, and that is for 2 minutes. Okay. So, I think that so from the PCR expression, then you can see that from the PCR experiments, if you can quantify that whether these certain specific genes related to the mature cell type is now upregulated or it is being quantified, then only you can confirm the cell differentiation process. Third one is the cell death. So, essentially if you go back to my earlier or you can recall if my earlier discussion, that a cell to be viable or cell to survive, cell has to receive different type of signal molecules A, B, C, and then if each of these or some of these signal molecules are removed or now absent in the cellular microenvironment, then cell has two options. Then either cell will undergo apoptosis, that is the programmed cell death, or in case cell receives some physical injury or tissue receives some physical injury, then it can undergo unprogrammed cell death, and this is called necrosis. The way apoptosis and necrosis can be distinguished, that is by morphological changes. So, apoptosis is a Greek word meaning dropping off. So, dropping off leaves, like the way leaves in certain specific season, they drops off from the plant. So, similarly apoptosis is a programmed and physiological mode of cell death, and that plays in tissue homeostasis, and it is also described as a genetically coded cell death program, which is morphologically and biologically different from necrosis or accidental cell death. So, how to quantify or how to identify this cell apoptosis process? In the cell membrane, you have the double lipid bilayer structure. In a live cells, when a cell is live, then you have specific molecules, protein molecules, the phosphatidylserine, which is always located towards the cytoplasmic space of the cell membrane. That means it is attached to the inner leaflet of the cell. When a cell undergoes apoptosis, this phosphatidylserine, which is shown here like a green sphere, this phosphatidylserine molecule will now be flipped towards the extracellular side. That means the phosphatidylserine molecule will now be switched to the outside, that outer leaflet of the cell membrane. And this, once it is located at the outer cell membrane, then there is certain fluorescent dye like Alexa fluor 488 or anisine. These can be tagged to the phosphatidylserine. So, this local tagging can be quantified by a technique called fluorescent activated cell sorter analysis or flow cytometry technique. Another thing that has been shown here. So, there is another fluorescent dye is called propidium iodide. So, this propidium iodide, it has a unique property, like it can intercalate with the DNA of a cell, and once it intercalates with the DNA of a cell, then it will fluoresce in the blue stained region, and then you can see that where the nucleus is located inside the cell. So, these two mode of the death, one is that apoptosis or necrosis has been summarized here in this slide. So, one is known as the very regulated process of cell death that occurs as a normal part of the cellular development, and it is called phosphatidylserine. It is known as the programmed cell death. Another one is unprogrammed or accidental cell death, which is characteristically morphologically different by membrane disruption, cell swelling and rupture often leading to inflammation. So, this is the cell morphological features during these two modes of death process. One is the necrosis. You can see all the intracellular substances are now coming out and the total destruction of the cell, and then apoptosis, which is essentially characterized by cellular shrinkage, also membrane blebbing here you can see, and nuclear condensation and fragmentation that you can see here, not that clearly, but it is that engraft dead cell, and this is a phagocytic cell. So, there is distinct morphological difference between necrosis and apoptosis, by which you can identify that when you see that there is no morphological change in the cell, when it is trying to adhere on a material substrate, it remains spherical. That means, you can well realize that cell is not willing to grow on this material substrate, and probably cells are not alive and cells are dead. Like in the cell division process, cell death processes also can be expressed by certain mathematical formulae, and this is one of the things that has been shown very generally in the cell death process, like rate of change of cell numbers with time dx by dt is equal to kx, where k is the rate of apoptosis. So, at any given point of time t, x t is equal to x naught exponential k t. So, this rate of apoptosis k essentially tells you that how fast the cell would undergo apoptosis. So, one of the things that I have mentioned in the last two modules is that fluorescence activated cell sorter analysis. So, this slide explains that what it means essentially. So, fluorescence activated cell sorter analysis is quite important because it allows single cell or one cell or single cell specific analysis, like it can allow the identification of the cellular fate processes for a single cell. So, you have a certain fluorescent stained cells which is made to flow through a column here, and once it goes through this column, then laser will be focusing on this specific laser will be focusing on these individual cells. Now, these red stained cells and black stained cells are showing that they have either in different sizes or they have a different cellular fate processes. Now, there is two type of scatter, one is called side scatter and one is called forward scatter. So, side scatter and forward scatter is shown here. So, side scatter essentially tells you that is size of the healthy or dead cells, size of healthy or dead cells, and the forward scatter, and side scatter essentially will tell you that what is the fluorescent and granularity changes. Now, once the cell goes through this fluorescent this is the FACS column, then if you induce certain or if you allow certain electromagnets to be placed there at the end of the column, then it will be it will enable you to sort the cells depending on what kind of negatively stained samples and positively stained samples. So, this important process can be very useful to quantify cell proliferation or cell cycle analysis or cell apoptosis on a single cell specific basis. Now, why this is unique in nature? Because all other biological all other biochemical assays, they essentially deals a given cell population where a large number of cells are present. So, one cannot therefore identify whether individual cell what kind of say what kind of cell fate processes they are actually at. So, this is again shown here in the side scatter and forward scatter, and this is a typical protocol that that a researcher follows while using the cell fate processes. Now, one of the thing that can be very useful for one to analyze is that how to quantify in a given cell population. Suppose you have a large number of cell population, let us say 10 to the power 3, 10 to the power 4 number of cells. We need to know whether that X percentage cells are apoptotic cells and then Y percentage cells are necrotic cells. Now, this distinction can be made by this fluorescence activated cell sorter analysis. Even you can also see that whether the cell is in the early stage of apoptotic or late stage of the apoptotic. So, what are the fluorescent dyes that you have to use? One is the Alexa fluor 488. So, this one will tag to phosphatidyl serine molecule as I have mentioned before. Propidamidite will intercalate to DNA double helix structure as I mentioned before. Now, what you can see here, this is the last slide before the end of this module. So, this is the 4 quadrants. So, Q1, that is quadrant number 1, here you have a very large expression of PI, but very small expression of lower expression of Alexa fluor 488. So, this is the necrotic cells. The live cells means it is stained with the PI, but the Alexa fluor 488 also stained, but it is in the lowermost quadrant here. Each dot in the fluorescent activated cell sort essentially represents the information from individual cell. So, the more number of dots in a particular given quadrant means more number of cells or larger cell fractions are present in that particular quadrant. If you go from this quadrant to this quadrant, that means cells are in the different stages of the cell fate process. So, this one is your live cells, this one is early apoptotic, then this goes to late apoptotic and finally necrotic. So, as from these simple things what you can see here, very few cells are in the late apoptotic and necrotic. So, more than 95% of the cells are in the live cells. So, that means that this particular cell population is largely alive with negligible fraction of the cells are in the apoptotic or necrotic stage. I think I will stop this model now.